Hello guys, welcome back to my YouTube channel, that is Biology at your fingertips, and today we are studying a very important topic for all the competitive examinations in which biology is a part, and the name of the topic is O2HB dissociation curve, that is oxygen hemoglobin dissociation curve, which forms many important questions. So I am going to emphasize on this particular question, that what can be asked from this particular article that we are going to learn today. So oxygen hemoglobin dissociation curve. Firstly, we have to know that HB can bind to 4 O2 molecules. This can be the first question, that how many oxygen does HB can bind to? It can bind to 4 oxygen molecules, that is 4 O2 molecules, and it provides our blood red color. The next question is, the element present in hemoglobin is iron, and the oxidation state of iron in hemoglobin is +2, which is a very important question not given in NCERT. So the oxygen dissociation curve is the plot between percent saturation of hemoglobin. What do you mean by saturation of hemoglobin? That how many oxygen are binding to a particular hemoglobin molecule. If it is binded to 4 oxygen molecules, it is called 100% saturated. If to 3, then 75% saturated, 50% saturated, 25% saturated, and so on. So it is the graph between percentage saturation of hemoglobin and partial pressure of oxygen. That what is the partial pressure of oxygen in mmHg? This is very important point, in mm mercury. So this graph is like this, and this particular shape of graph is known as sigmoid. This forms another important question from this article, that this particular graph, this shape of graph is known as sigmoid. And why it is sigmoid? That when a single O2 molecule bind to HB, it is quite difficult to bind, but this oxygen facilitates the binding of another O2 molecules, so this O2 molecule can bind easily. 2 O2 molecules facilitate the third oxygen molecule, so this oxygen molecule will bind even more easily, and so on. So the graph is sigmoid, and when we increase the partial pressure of oxygen, after a certain time the graph saturates, because all the 4 positions are already occupied by different oxygen molecules in a particular hemoglobin molecule. So the graph saturates. Now what are the factors that are affecting oxygen hemoglobin dissociation curve? PCO2. We know that the CO2 has 20 to 25 times more binding capacity to hemoglobin than oxygen. So when we increase the partial pressure of CO2, then the graph will shift to right, because when CO2 increase, the oxygen will not be able to bind hemoglobin. After that, if we increase temperature, the oxygen will again not possible for oxygen to bind hemoglobin molecule, so it will again shift the graph to right. When we decrease pH means we are increasing the acidity of a solution. So when we make the solution acidic, the graph again shifts to right, because the oxygen will not be able to bind HB molecule. The PO2, the more the number of PO2, more will be the binding of it with HB. So when we increase O2, the more HB molecules get saturated with O2, so the graph will turn to left. PCO, we know that CO has 250 to 300 times binding capacity than oxygen with hemoglobin. So when we increase the amount of PCO, the graph will turn right. Fetal hemoglobin. The oxygen carrying capacity of hemoglobin in fetal hemoglobin is more than our normal hemoglobin, because when the oxygen is getting circulated via blood, it is more important to reach it to the fetal blood. So fetus has much interaction with oxygen than normal blood, than normal hemoglobin. So when fetal hemoglobin, we talk about fetal hemoglobin, then the oxygen dissociation curve will bend towards left side, because more oxygen are getting binded to the Hb molecule, or more affinity is shown by oxygen to bind to Hb molecule. So a simple trick to memorize all the factors that are affecting oxygen dissociation curve can be: all the factors that are breaking the HbO2 bond or promoting their dissociation will turn the graph towards right direction, this direction, and all the factors which are promoting HbO2 will turn the graph towards left side. So we have talked about each and every factor that is affecting. For fetal hemoglobin, the hemoglobin level in fetus is much higher, that is 23, which is much higher than the normal man or normal women. So the affinity of the foundation of oxygen to hemoglobin will be much more in fetal hemoglobin. This forms a really important question in many PG examinations such as IIT JAM. So we have to note it down. So these are the factors affecting oxygen hemoglobin dissociation curve and all about oxygen hemoglobin dissociation curve. In normal, 100 ml of oxygenated blood contains 5 ml of oxygen. This is a data which is given in NCERT book. So this is all about oxygen hemoglobin dissociation curve and all the factors which are affecting this curve. So thank you so much guys for watching this video. If you really like this video, then press the like button, and if you are new, then please subscribe to my channel. Thank you so much again.